Welcome back to the Deep Dive. Today we're tackling one of those topics that it really sits right at the intersection of biology, anxiety, and, well, a bit of mystery. We're talking about the biological clock.
It's the phrase that launches 1,000 frantic Google searches.
Absolutely. And we all know the headline, right? If you're a woman, your fertility drops as you age. The risk of miscarriage goes up.
Right.
IVF becomes this grueling numbers game with just diminishing returns. We sort of accept this as a universal truth, like gravity.
Or taxes.
Or taxes. But if you actually press a doctor and ask, okay, but why? What is physically breaking down inside the cell? The answer is usually pretty vague.
You usually just get the term egg quality.
Right. Poor egg quality.
Yeah.
Which feels like a bit of a cop-out. It's like a mechanic telling you your car won't start because of car quality issues.
That doesn't help much.
No. Is it the spark plugs? The battery? I mean, what are we talking about here?
That's a fair criticism. For a long time, egg quality really was a black box. We knew the outcome. Older eggs don't make babies as easily, but the molecular check engine light was just flashing. We didn't have the code.
Well, today we are finally popping the hood. We've got a fascinating paper here from the Max Planck Institute, led by Melina Shu and her team. The title is Restoring Shugoshin 1 Reduces Chromosome Errors in Human Eggs. And honestly, this paper feels like a sci-fi prequel.
It really does. Because they didn't just describe the damage, they found a specific protein that acts as a kind of guardian for our DNA.
Okay.
They figured out why it abandons its post as we age, and then, and this is the part that blew my mind, they found a way to bring it back.
It sounds like they found a rewind button for the egg's internal machinery.
It is the closest thing to a rewind button I have seen in this field in years. I mean, it really challenges the idea that aging is just inevitable, irreversible wear and tear.
So our mission today is to understand this guardian spirit, Shugoshin 1. We need to understand the crime scene, what actually happens in an aging egg.
What causes the errors?
Exactly. And then we'll look at the rescue mission. How did they fix it?
Let's get into it.
Okay, so to understand the solution, we have to understand the unique pressure these cells are under. Walk us through the lifespan of a mammalian egg. Because it's not like a skin cell, right?
Oh, not at all. No, the oocyte, the egg, it plays the longest game in biology. A woman is born with all the eggs she will ever have. In fact, they're formed when she's still a fetus.
Which is always a trippy concept. The egg that made me was inside my mother when she was inside my grandmother.
It's a biological Russian nesting doll. So these eggs form, and then they enter a state called prephase eye arrest. Think of it as suspended animation.
A pause button.
A pause button. They just sit there. Waiting.
Decades.
Exactly. For 30, 35, 40 years.
Yeah.
Now, imagine holding a plank position for 40 years. That's essentially what the chromosomal machinery inside the egg has to do.
Wow.
And the biggest risk during this long, long hold, something called aneuploidy.
Aneuploidy. Let's define that clearly because it's really the villain of our story.
It is. The simple definition is the egg ends up with the wrong number of chromosomes.
The math is wrong.
The math is wrong. Humans need 23 pairs if the egg has an extra or is missing one. Well, if that egg gets fertilized, the embryo usually just can't develop. This is the main cause of miscarriage and IVF failure in older women.
So it's not that the egg can't be fertilized, it's that the genetic instruction manual is just garbled.
Perfectly put. And the paper identifies a specific mechanism for how this happens. They call it PSSC.
Premature sister chromatid separation.
It's a mouthful.
It is a mouthful. I always visualize it as two siblings all holding hands in a crowded mall.
That is the perfect analogy. Before an egg divides, it duplicates its DNA. So you have two identical copies, sister chromatids.
Okay.
They're literally tied together. They have to hold hands tightly for those 40 years. They can only let go at the exact split second of fertilization.
And PSSC is when their grip slips too early.
Correct. They let go before the signal, they drift apart, and when the cell finally divides, one daughter cell might get both sisters and the other gets none.
And that's the error. That's aneuploidy.
That's it. So the integrity of the whole genome relies on the glue holding these hands together.
Which is a protein complex called cohesion.
Yes. And here's the scary part, and this is unique to eggs. This cohesion glue is loaded onto the chromosomes when you are a fetus. And it is never replenished.
Wait, never? The body doesn't make new glue.
Not for the eggs. The glue you have at 40 is the same set of molecules you synthesized before you were born. There's just no turnover.
That seems like a massive design flaw. It's like trying to hold a house together with 40-year-old duct tape.
I mean, eventually it's going to dry out and crack. And that was the prevailing theory for a long time.
Just entropy.
Entropy wins, the tape dries out, things fall apart. But this study asked a sharper question. Is it just the glue failing? Or is there something that's supposed to be protecting the glue that's falling asleep on the job?
Enter the guardian.
Enter SGO1, Shugoshin 1.
I looked this up. Shugoshin is Japanese for guardian spirit. Scientists really nailed the branding on this one.
They really did. It's appropriate. Shugoshin 1 sits at the centromere, the pinched center of the chromosome, and its sole job is to protect that cohesion glue.
Protected from what, though?
From the cell's own machinery. There are enzymes in the cell, like one called separase. Think of it like molecular scissors that are just itching to cut that glue.
To let the cell divide.
Right. SGO1 is the bouncer. It stands in front of the glue and says, "Not yet. Keep the scissors away."
But SGO1 doesn't do this alone, right? The paper mentions it recruits a partner.
Exactly. It recruits a molecule called PP2A. If SGO1 is the bouncer, PP2A is the shield he's holding.
Okay.
SGO1 grabs PP2A and positions it right over the glue, specifically a part of it called REC8 to block those scissors.
So we have the glue, Cohesion, the guardian SGO1,
Yeah.
and the shield, PP2A. Now here's where the Max Planck team found something new. I always assumed this was a static system.
Like a lock on a door.
Yeah, you lock it, you leave it for 30 years. But they found it's actually incredibly active.
This was a surprise. They found that in mice eggs, the centromere, that center part of the chromosome, is actively being transcribed.
Transcribed. So reading the DNA to make RNA. I thought chromosomes were supposed to be quiet during this whole arrested phase.
We all did. We thought they were dormant. But it turns out there's a low-level constant reading of the DNA right where the guardian sits. It's like a keep-alive signal.
And why does it need that?
Because SGO1 is unstable. It doesn't stick around forever. It degrades and needs to be replaced or, stabilized. So the cell needs to constantly read the instructions to keep the SGO1 protein active right at that spot.
So it's not a lock, it's a juggler. If you stop paying attention, the balls drop.
Exactly. And they proved this with a really elegant experiment. They took young, healthy mouse eggs.
Prime fertility.
Right. And they added chemicals like triptolide to stop that transcription. They basically hit the mute button on the DNA reading process.
And what happened to the guardian?
It vanished. Without that active signal, SGO1 just disappeared from the chromosomes. The shield, PP2A, fell off, and immediately the chromosomes started drifting apart.
PSSC errors skyrocketed.
They did. So by turning off that signal, they artificially aged these young eggs. They made a 12-week-old mouse egg act like a geriatric one.
Wow. Just by stopping that reading process.
Precisely. It confirmed that the guardian needs constant maintenance. It's an active process.
Okay, so that establishes the mechanism in a healthy egg. Now let's look at the real villain, natural aging. They took grandma mice, about 60 to 65 weeks old.
Which is elderly for a mouse, yeah.
What did the crime scene look like in those eggs?
It looked like a system in collapse. First, they confirmed that the reading of the DNA, that transcription, was much, much lower. The signal was fading.
So the keep alive ping was getting weaker.
Much weaker. And as a result, SGO1 levels were plummeting. There was barely any guardian left.
And no guardian means no shield.
No shield. The glue was exposed, and the chromosomes were literally coming apart. They measured the distance between them, and they were just loose.
Okay, now I want to get to the twist in the paper. Because my instinct would be, okay, the signal's too quiet. Turn up the volume.
Right. The logic suggests if the DNA isn't making enough RNA instructions, just inject the RNA.
They tried that, right?
Yeah.
They injected something called major satellite RNA, which is the specific message produced there.
They did. And here's the fascinating nuance. When they did this to the young eggs that they'd artificially damaged with the chemicals, it worked.
It fixed them.
It fixed them. The guardian came back. Everything was fine.
Because the machinery was still young and ready to go.
Exactly. But, and here's the twist, when they injected that same RNA into the naturally aged eggs,
Let me guess.
nothing happened. It failed.
Why? Why would it work in the young damaged egg, but not the old one?
This is the key insight of the paper. The aged eggs weren't just missing the signal. They were missing the ability to translate that signal into the protein. The factory itself was broken.
You can shout instructions at a factory all day. That's the RNA. But if the assembly line is shut down, you aren't getting any product.
That is a crucial distinction. It wasn't a communication problem. It was a supply chain problem. They were just depleted of the SGO1 protein itself.
Yes. The whole environment of the aged egg was just too tired to build a guardian from scratch, even with the blueprints. The protein level was the limiting factor.
So if the factory is broken, you have to import the finished goods.
Welcome to the rescue mission.
They decided to bypass the factory floor. So instead of the RNA signal, they injected mRNA that codes directly for the SGO1 protein.
They essentially airdropped the Guardian into the cell. They injected SGO1 mRNA, which is a direct template. The cell's general machinery could easily grab this and just turn it into protein.
Flooding the egg with fresh Shugoshin 1, and
It was like watching a time-lapse in reverse. The SGO1 levels shot back up to youthful levels.
yeah.
PP2A, the shield, was recruited back to the chromosomes, and the error rate, the PSSC, it dropped drastically.
How drastically are we talking? The error levels became indistinguishable from young mice.
Wait, really? Indistinguishable?
Statistically, yes. They effectively reversed the chromosomal instability. The old chromosomes stopped drifting and held hands tight again.
That's incredible. But, being a good skeptic, was it really the SGO1? Or did they just shock the egg into waking up?
That's a fair question. They ran rigorous controls to prove it was specific. First, they tried injecting SGO2. It's a cousin protein.
Did that work?
No. SGO2 is important, but it couldn't do the specific job. It failed to rescue the eggs.
Okay.
Then they also injected a mutant version of SGO1. It was engineered so it couldn't grab the PP2A shield.
A Guardian with no shield.
Exactly. And that failed too. This proved you need SGO1 specifically, and you need it to be able to physically grab PP2A. It's a lock-and-key mechanism.
All right, mice are great. But the leap to human oocytes is usually where these kinds of amazing discoveries fall apart.
Often, yes. Mouse and human biology have significant differences, especially in reproduction. But this team, they went the distance. They collaborated with fertility clinics and obtained human oocytes donated by women undergoing ICSI.
So these were eggs that maybe failed to fertilize or were immature?
Exactly. Donated for research. They had eggs from women under 35 and women over 35.
First step. Confirm the problem even exists in humans.
And it did. The pattern held perfectly. The older human eggs had significantly lower SGO1 levels and much higher rates of chromosome errors. The biology is conserved.
So then they tried the therapy. They microinjected the SGO1 mRNA into the human eggs.
And this is the statistic you need to remember. In the untreated group of older eggs, 53% of them had chromosome separation errors.
53%, so more than half.
More than half. It's a coin flip, a bad coin flip every time.
Right.
Now, after the SGO1 treatment, that error rate dropped to 29%.
From 53 to 29, that is nearly a 50% reduction in errors.
It is huge. In the world of fertility, where we fight for single percentage points of success, an effect size that large is, well, it's startling.
It doesn't eliminate errors, but...
No, 29% is still significant, but it fundamentally changes the odds.
And the way they tested this was clever, too. It wasn't just comparing one group of women to another.
No, they used it within donor design. This is the gold standard. They took eggs from the same woman, treated half, and left the other half alone as a control.
So you can't blame genetics or lifestyle. You can't say, oh, donor A just ate more kale than donor B.
Exactly. Same person, same history, same genetics. And in almost every single case, the treated eggs from the same woman were better than her untreated ones. It isolates SGO1 as the causal factor.
This feels revolutionary. I mean, currently, the strategy for older women in IVF is basically just a numbers game. It's volume.
It's a selection strategy. We stimulate the ovaries, get as many eggs as possible, fertilize them, and then we screen them genetically to throw away the bad ones.
So we admit the eggs are broken, and we just try to find the one or two that aren't.
Yes. But this suggests we could actually repair the hay instead of just searching for the needle. It shifts the paradigm from inevitable decline to treatable deficiency.
It suggests the errors aren't just structural rot. They're regulatory failure.
Yeah.
And you can fix those.
Exactly.
Now, before listeners start calling their clinics demanding SGO1 injections, we need to be realistic. This isn't happening tomorrow.
No, absolutely not. There is a huge gap between improving chromosome structure in a dish and producing a healthy baby.
What are the risks? What are we worried about?
Well, we don't know if flooding an egg with SGO1 has long-term effects on the embryo. SGO1 is a powerful regulator of cell division.
So too much of it could be a bad thing later on?
It could be. Does it mess up the embryonic cells dividing? Does it cause cancer risks? We just have no idea yet.
Plus, microinjecting every single egg is technically difficult and expensive.
True. But the proof of principle is what matters here. It tells us that the egg has the capacity to be rescued. It's not a lost cause.
It changes how I view aging entirely. We usually think of aging like mechanical wear and tear. The piston rings are worn out, the tires are bald. You can't unwear metal.
That's the entropy view. Things fall apart.
But this research suggests that, at least in this one case, the car isn't broken because the metal is worn. It's broken because it ran out of oil. And if you pour the oil, the SGO1 back in, the engine runs smoothly again.
It frames aging as a maintenance issue. The blueprint is still there. The machinery is just dormant or undersupplied.
Which leaves me with a final, slightly provocative thought.
Go for it.
We tend to compartmentalize aging. My knees hurt, my eyes get worse, my eggs get old. But if the guardian of the genome in eggs is essentially just a missing protein problem, what else is?
That is the billion-dollar question.
Are our neurons losing their memory just because they're missing some stabilizer protein? Are our hearts getting weaker because of a missing enzyme?
It's entirely possible. The oocyte is arguably the most fragile cell in the body because it has to last so long without dividing. It might just be the canary in the coal mine.
So if we can refill the tank for the oocyte,
Why not the others?
it's a hopeful thought.
Yeah.
Maybe we aren't decaying so much as we are just running empty, and tanks can be refilled.
Let's hope the mechanics keep working on it.
We will be watching. That's it for this deep dive into the guardians of the genome. Thanks for listening.